Good afternoon and welcome. 30 years ago, I started Friends of Cancer Research because of a profound personal loss. It is my clear belief that if the right people in the room together, researchers, patients, FDA, and Congress, we can do more than any one of us can do individually. This is really essential. Today is exactly that. Biomarkers are not an abstract concept. They are tools that get the right treatment to the right patient faster. There is nothing more important to a patient. And now I want to take a moment to introduce someone I have enormous respect for. Senator Tim Kaine has been one of the most thoughtful and effective champions of FDA and for patients. Right now, the FDA's capacity and long-term direction are serious issues that must be addressed. His voice carries real weight, and we are honored he is with us today. Senator Kaine, thank you for your leadership and for opening this briefing. The floor is yours.
Hey, everybody. Senator Tim Kaine here. Let me welcome you all to the Friends of Cancer Research virtual Hill briefing called Strengthening FDA's Biomarker Qualification Program. Now, the Food and Drug Administration is the world's gold standard for drug evaluation and approval. A strong FDA is what makes the U.S. the leader in biomedical research and innovation. When the FDA functions well, patients get access to safe and effective treatments. But when it doesn't, patients have the most to lose. It's because of advocates like you all here today that Senator Collins and I passed the bipartisan Modernizing the Accelerated Approval Pathway Act, using the latest FDA user fee reauthorization cycle. Our bill improved upon the accelerated approval pathway by encouraging FDA to set benchmarks for drug development and strengthen transparency throughout the process. This was a step in the right direction, but we know that more has to be done to support patient access. Congress has a responsibility to ensure FDA has the resources, the staff, and the scientific infrastructure necessary to get the job done. And this isn't a partisan issue, it's a patient safety and access issue. Every 5 years, Congress gets an opportunity to review the FDA's user fee programs to make sure we can improve safety, reliability, and efficiency, making sure the United States remains the world leader in cutting-edge drug development. This coming reauthorization is going to be particularly important. The FDA has seen tremendous staffing cuts and turnover. Currently, it doesn't even have a confirmed director. So I'm deeply concerned that the FDA is at risk of losing the trust of the American people. Many of the FDA's choices have appeared political in nature, like overruling career scientists to reject an mRNA vaccine application. The FDA has to be guided by the best available scientific evidence and guided by its seasoned career scientists. As we approach the coming user fee reauthorization, it's critical that Congress thoughtfully evaluate what's working and what's not, so that we can ensure patients have access to the most safe and the most effective drugs possible. I'm committed to making sure that patients with rare and ultra-rare diseases get fast access to treatments that meet the FDA's rigorous framework for safety efficacy. The way to do that is by listening to our career scientists, not overruling them, and improving our user fee programs to ensure they have the support they need to make timely and unbiased decisions. And you have a job to do as well. The voices of patients, experts, and advocates, such as Friends of Cancer Research, are really important. My staff and I are so grateful to work with you on all these issues. Dr. Jeff Allen, thanks for the chance to talk today. Thank you to the whole Friends of Cancer Research team, and my best wishes to all the attendees today for a productive session.
Thank you, Senator Kaine, for joining today's discussion and for your continued leadership to advance important programs like the FDA Accelerated Approval Program. Today's briefing will focus on a topic that is key to the Accelerated Approval Program at FDA and many other aspects of ensuring efficient, rigorous, and science-based clinical trials. Specifically, we'll be discussing the FDA Biomarker Qualification Program and its important role in facilitating scientific advancement in the development and safe and effective medicines for the people who need them. Biomarkers are measurable characteristics in the body that can be indicators of healthy processes, abnormal function, presence of disease, or response to treatments. They are measured by a wide variety of tools such as physical assessment, molecular or genomic testing, or imaging technologies. The results provided from biomarker evaluation can help with disease detection, inform treatment decisions, monitor safety, or indicate whether a treatment is working. This enables biomarkers to be used to optimize research in a variety of ways, including improve clinical trials by identifying the right patients for specific treatments, add efficiencies to drug development, including for rare diseases, provide the basis for accelerated approvals as early measures of benefit, and enhance regulatory decisions by providing additional information about drug safety and efficacy. Given the opportunities presented by rapidly advancing science and technologies, a clear pathway for developing and validating biomarkers is needed. Several years ago, as part of the 21st Century Cures Act, Congress recognized the need for researchers to work with regulators as part of developing new biomarkers by establishing the FDA Biomarker Qualification Program. The goal was to enact a set of processes that would provide a predictable path to generate the evidence needed to support biomarker development, and that those validated biomarkers could be used across drug development programs without repeated review. A three-step process was established, initiated first by a letter of intent submitted by a company or research consortium to the FDA, followed by a qualification plan, which serves as the experimental plans for developing the necessary evidence, and finally, a full qualification plan containing the results supported the potential use of a biomarker. Recently, we published a landscape assessment to help characterize the experience with the Biomarker Qualification Program. In this study, we found that while 61 research projects have been accepted by the FDA to the BQP, only 8 biomarkers have been successfully qualified. Of the 8 that have been qualified, 7 of those programs were submitted prior to the establishment of the BQP in the 21st Century Cures Bill and grandfathered into the program. The past decade of experience indicates that few biomarkers reach qualification, even though they were approved for entry by the FDA, and many programs stalled in the process. In addition, review timelines are lengthy and unpredictable, and it can be unclear what evidence to support biomarker qualification is expected by the FDA. Since the time of the study, FDA has qualified additional biomarkers through the BQP and sought to make positive process adjustments. But given the experience to date, we convened an expert working group of researchers, regulatory and policy experts to develop a set of external recommendations that stakeholders felt would enhance the biomarker qualification program for the future. The full set of recommendations to modernize the BQP are described in a comprehensive white paper that is now available and includes areas where Congress could help provide modifications and further support this important program. The recommendations fall within 3 main categories. First, establish predictable processes and clear accountability mechanisms. Recommendations in this category recognize the need for resources to support timely review and feedback, adherence to predictable timelines, and reporting on progress of program applications. Number 2, strengthen institutional and regulatory alignment to clarify how qualified biomarkers can support subsequent clinical trials and overall drug development programs. This includes processes to readily enable expert FDA staff participation in biomarker development and review to ensure continuity. And third, modernize the BQP to align with evidentiary maturity. Here we propose a new tiered approach to biomarker development and validation that enables risk-based assessment of the biomarker. This would help recognize that evidence supporting the use of a specific biomarker will accumulate over time. New processes are needed to clearly characterize the level of evidence regarding a biomarker, including additional research conducted, and then to modify its appropriate use as needed. The recommendations developed with the working group will be expanded upon during today's panel discussion with representatives from the group. I want to thank all of the working group members for their time and expertise in developing the white paper, and I hope that all of you will have the opportunity to review the recommendations in greater detail. The BQP is an important program to support future clinical trials and development of new medicines for the people who need them most. We look forward to working with many of you to explore opportunities to move these ideas forward. I'd now like to turn things over to our moderator for today's discussion, Dr. Rasika Kalamegam from Genentech. Thank you.
Thank you so much, Jeff, for setting the stage and highlighting the criticality of biomarkers and other drug development tools to modern drug development. It's my pleasure to continue the discussion and focus on how the qualification program can be strengthened to modernize drug development. I'm joined today by our panelists, Steve Hoffman from the Foundation for the NIH, Mike Montalto from Amgen, Lauren Oliva from Biogen, and Katie Wack from PathAI. Welcome, everyone. And we've all been working together for a few weeks now on this white paper. And today it's just a chance for us to tell the world about why biomarkers and drug development tools matter and what can we do to improve the process so we're actually leveraging these tools as we bring newer and better drugs to patients. Jeff just talked about how important these tools are. So I just want to start off by setting the stage and asking, why does qualification matter? And what does it mean in practice when the qualification process does not keep pace with discovery science? I'm going to start with you, Mike. How about you kick us off?
Yeah, great. Okay, thank you so much. And thanks for having me on the panel. So first let's just start with just how important biomarkers are to the drug development process. Everything from selecting patients to stratifying to understanding how our drugs work, and from a pharmacodynamic perspective, that is to say that the mechanism of action that you think that you're hitting, you're actually hitting, regardless of efficacy. That can be super important. And all of those pieces of data that we get from biomarkers helps us to make faster decisions and ultimately get our drugs to patients more quickly. So there's a huge public interest in getting drugs to patients more quickly, and biomarkers is a really important part of that. A qualification pathway is a regulatory pathway by which we are able to all sort of agree that a particular biomarker or a particular method to use a biomarker has met certain evidentiary standards, and therefore, as an industry, we can use it, and we don't have to re-meet those standards every time we want to use it, and that saves a tremendous amount of time. So the concept of the biomarker qualification program is beautiful, because I really do think that the intent is to try to accelerate the use and adoption of biomarkers in drug development. So it's super important, and when the regulatory process doesn't work very well, we sort of fall down a little bit on that promise that we think that biomarkers can help. So that's sort of the gestalt of it, so to speak. Yeah.
Yeah, and all of us see value in the qualification pathway, not just for biomarkers, but for drug development tools like COAs and PROs and PERFOs and so on. But we've all run into practical challenges when we've tried to use it. The issue is the time, the resources, and ultimately the uncertainty involved. It's exactly what you said, Mike. It's about having regulatory certainty that once you've qualified something, you can use it in your clinical development program with confidence, and we don't have that confidence. Friends has done some research showing some of this data, and we have as well. Friends found that about 13% of biomarkers have passed the bar through this pathway, and we looked at COAs and we only found that about 8% of them met the bar. So given this track record, if you had a biomarker or a drug development tool that could benefit from broader regulatory acceptance, would you pursue the qualification pathway? And maybe why or why not? And what would you need to change it to make it worthwhile? I'll start with you, Lauren, and then I'll jump over to you, Katie.
Rasika, no, Mike summed it up pretty perfectly in terms of all the opportunities, and really we do see so much value in the pathway. So certainly I would say we would consider the biomarker qualification pathway, but selectively. So we see it as a very important pathway to build that shared regulatory confidence, particularly, as you mentioned, where that broader reuse across programs is possible and will create efficiencies. Today, what I will say is that much of the confidence as we see it is built through some of the early and ongoing regulatory dialogue through the IND pathway. So from our experience, that early and transparent alignment with FDA about how the biomarker will be used and what that uncertainty is intended to address in terms of those conversations in a particular program is often the most effective way to support development and avoid some of the downstream work. And I think one of the key key pieces of it is the data availability. That's a key driver for which pathways are feasible. As we've talked about in the white paper, there's a few different pathways, and qualification generally does require some existing and robust cross-program data sets, whereas the IND pathway does allow for evidence to be generated and applied in a more iterative manner across the development program as the scientific understanding evolves. That's particularly relevant in some of the areas that we work in. So in rare diseases and when you're using adaptive trial designs, as we often do for many of our development programs, there may be limited data or high heterogeneity within the patient population. And that makes the current qualification paradigm somewhat difficult to apply at times. So often sponsors will kind of look to the IND pathway to build on and apply that biomarker evidence in real time. So in practice, we do tend to use that pathway, as I mentioned, first, and often then consider qualification more selectively when there's that broader application. And maybe just one point to add, to make qualification a bit more impactful, we do really feel like the focus should be on better aligning that pathway to how biomarkers are actually developed. So that means reflecting how evidence matures over time and strengthening the linkage between that development-based use, as I talked about, so that confidence can build more iteratively. So thinking about expanding the context of use, scaling from kind of a specific context of use into other areas, whether that's other disease areas or other kind of contexts where you might use that biomarker in that particular patient population. And also leveraging prior knowledge. So where work has been done in other programs where you might be able to leverage and sort of not be restarting and reestablishing each time.
Yeah, and just to break it down for our audience that may not be as familiar with these acronyms, when you say IND pathway, you mean the way in which we all file our applications for a specific drug to be approved by the FDA. And when we speak about qualification, we're talking about a product agnostic way in which a biomarker can be certified as being acceptable to the FDA.
Right. Thank you, Roscoe.
No worries. That's why I'm here. So, Katie, you just recently went through the qualification process. And so based on your very fresh experience, maybe speak to us a little bit about the pros and cons. What did you find having gone through it so recently?
Yeah, I would be happy to. So we study many different indications and develop tools that are very helpful for various contexts of use. So I think entering this program, it was the first tool of its kind. And so we were really helping to establish what levels of evidence would be needed for analytical validation. And then importantly, it's not just the tool itself, but the workflow in which the tool is used in a trial that must be clinically validated. And so as we were establishing those, there were many other use cases that I think we would love to have entered into the BQP program to explore. However, because of the sort of uncertainty with timing and lack of mechanism to evolve with evolving technologies, evolving clinical trial populations over time, we took a more conservative approach and focused on this one use case. If we could align the operations of this program more so with product life cycles with an eye towards defining how we will evolve as some of these technologies evolve and as some of our tools evolve, I think we would join in many areas. There are many drug development tools that could benefit from some of our technology, but we would need a more predictable structure in order to enter the program for these projects.
Yeah, so in other words, if you could somehow have parallel tracks that were more in sync, you could actually use these tools in a timely manner to advance drug development.
Yes. And even to extend across programs, for instance, for similar biomarkers that may have a similar design for analytic validation to show accuracy and precision, if we knew those could be applied across appropriate, relevant other biomarker programs, we would certainly apply those if we could do so in an efficient manner.
So we have the will, we just need the way.
Yes.
I'll step back and say many biomarkers go through a journey, which is very similar to drug development. There's the discovery phase, there's the validation, and then the regulatory acceptance, and then employment in clinical development programs. And when Congress conceived of the pathway, one of the most attractive aspects was that it was meant to bring together industry, academia, patient groups, and each stakeholder could contribute to this joint effort. So, Steve, I'm going to turn to you because you have run so many of these collaboratives. You bring together these stakeholders because you are a nonprofit and your goal, your aim is to advance the science because you're not actually developing the product, the drug, in other words, but you are developing the tools that then help develop the drug. So based on your experience, please tell me, what does the level of uncertainty around qualification mean in practice for collaborative biomarker development?
No, I again appreciate, like others, the opportunity to be involved here. And your questions and the sage advice of my colleagues are really spot on. And these collaborative partnerships that FNIH puts together, consortia alliances, are inherently resource intensive. They require long-term commitments and coordination between, as you said, the academic sector, the pharma sector, regulators, patient groups. And really, when we focus more on what you had talked about, the biomarker qualification program, and in a product agnostic type of way, to put those pre-competitive partnerships together to forward biomarkers, clinical tools, endpoints, and validate them for a way that they can be adopted and used in a standardized and a harmonized way. Where we see unclear evidentiary thresholds, inconsistent interpretations of some of that evidence across the different review divisions of FDA is challenging. How we approach CDER versus CDRH or the other divisions, and having a consistent voice and message back and forth to them and from them is really challenging for what constitutes success in qualification, and how that success will translate reliably downstream into regulatory decision-making, particularly in drug development. So this uncertainty is compounded by unpredictable timelines and feedback cycles, which can extend some of these qualification efforts for years. We don't have the money to do that. You lose a lot of momentum from the existing partners because they don't understand the commitment and the time to take something from adoption and use within their trial or that they're working on versus when I need to really move this forward with benefit to qualification. So these challenges point to that gap and to the intended role of the qualification and the shared scalable regulatory tool in operational reality and how we're going to use it. And so these are the challenges that we see in the partnership, both in the funding and financial shared resources, but we lose that momentum and need and understanding. Is that really needed for the drug development to accelerate all parties, raise all boats, or is it kind of a nice to have instead of a must have in that scenario?
Yeah, this really resonates because collaboration is a wonderful word that we all like to throw around, but the collaboration must have an endpoint, and it should result in something that actually benefits all of the collaborators. And what I'm hearing you say is it's not that people aren't willing to share their resources or come to the collaboration. It's that they've tried multiple times, and the lack of the return on investment is now a disincentive to attract more people to come to collaborate.
Yeah, I would agree. And to follow on, certainly it's a two-way street. There are mechanisms that FDA has where the consortium or the collaborative needs to bring forward and interact early and often with mechanisms that FDA has on board, where we have seen success, where we've reached full qualification on a surrogate endpoint and osteoporosis and a biomarker panel and kidney safety. Those were long-term, we're talking about 10 years to get through that process. Very difficult to project, budget, plan, keep the momentum for that type of a sustained effort for that long period of time.
Yeah, we're almost at the halfway mark. So I'm going to pivot into solution mode because we just talked about all of the problems. So Mike, I'm going to go to you. We learn, qualification is step one. Once you have something qualified, then you have to apply the biomarker in a clinical development program to really realize the fruits of the investment in the form of a drug that's approved. We learn an enormous amount when the biomarker itself is used in clinical development, and usually when we reuse it, when we use it in another program, we learn more. So the question to you is, are we fully leveraging these learnings? Are we translating it into broader regulatory use?
Yeah, I mean, I don't. So the short answer is no. We probably don't. I don't think that there's a concerted way to do that. I think that, when we see this happen kind of retrospectively, where large meta-analyses are done, and we sort of go back and try to get at all of that data and see sort of what the data is telling us. I don't think that we do that proactively enough across programs and across sponsors. I certainly think part of this qualification, refresh could be a reuse of the data, a kind of applying it to, as Katie was mentioning before, if you wanted to go into new indications, kind of applying those learnings in such a way across these programs that we could accelerate the next step in that process. So it does wind up, especially if it's not even part of the qualification, as Lauren was saying, if it's part of an IND, then it really becomes isolated. But I think if there were in many ways incentives and predictability that data was going to be leveraged and used to continue to expand the use of the biomarkers, I think that there would be a lot of interest in doing that. And Rashka, I want to say one other thing, and I meant to say this earlier as Katie was talking, because it's a nuance that may be lost on us. She mentioned emerging technologies and evolving technologies as part of this discussion. I think it's important that we also realize, and I'm sorry to go off script here a little bit, that AI is really changing the game for us in the biomarker space. So we talk a lot about the biomarker qualifications and all the different contexts of use, which is the biology of that biomarker. But applying AI to existing biomarkers, and PathAI was a real leader in this, and applying it to aim to NASH and the histological endpoints of that. But we see that for things like IBD with histological remission endpoints, for RECIST in cancer, which applies to so many solid tumors. Just the application of AI to those spaces would accelerate drug development and the use of those biomarkers for patients. And so we should include in this discussion the application of AI and qualifying an AI tool for existing biomarkers that we have today. I'm not sure that the current qualification pathway kind of envisioned that when it was established. So there would be kind of a whole host of other things that we can think about. So I didn't want to lose that point because it was sort of something that Katie brought up earlier. So
yeah, if I were to sum up what you were saying, both of your points touch on leveraging prior data, right?
Yeah.
Leveraging prior knowledge. So either we're leveraging prior knowledge to expand the context of use of the biomarker, or we're leveraging prior knowledge not to establish the validity of a biomarker itself, but figuring out how do we better measure it, how do we better capture it, because we have better technological tools today compared to what we had 20 years ago. And that's
exactly
the point about RECIST or IBD biomarkers, et cetera.
Exactly.
I'm going to go to Lauren. If I take a step back, I work in a company that develops drugs. You do too, Lauren. Qualification was really intended to de-risk the use of innovative tools and provide a regulatory signal that both sponsors and reviewers can rely on. So in practice, we've just laid out all of the reasons why this is not happening, and there isn't that level of confidence to go through qualification. So what do you think needs to change for this pathway to become more useful?
The $100 million question, isn't it?
I think billion is what we're going for now. We've moved past million.
There we go. True. But yeah, no, I think I kind of jumped into solution mode in the kind of the challenge portion of our conversation. But as you summed it up, in practice, qualification, I don't think the aspiration has been fully realized. I think that's why we all came together to have these conversations, to say we support this, we can get behind it, we're happy to share our data, but how do we make this more fit for purpose so that the uptake is really realized? I mean, Steve talked about the surrogate endpoint that they successfully qualified, but as I recall, I think there's been only one surrogate through BQP in the history of the program. So there really is a lot of opportunity, and I think that's why we're having this conversation. So, I mean, in terms of that regulatory confidence and realizing it, I mentioned in my earlier response that ongoing dialogue with FDA and kind of the integration of the different sources of data, whether it's clinical or mechanistic and all of that, is a very important piece of moving the biomarkers forward. In our experience, one experience I'll maybe mention is around our QualSADI program, which is for SOD1 ALS. We were looking at plasma neurofilament light for ALS, and I think it's a good example because neurofilament light, it really started as an exploratory biomarker for our program. It was in early stage studies, and we very deliberately advanced it over quite a number of years. And as the evidence matured, we were able to link it to the biology, to the target engagement, to the clinical outcomes, all of those very important pieces of what it takes to move a biomarker from informative to decision making. And it did ultimately, as I mentioned, support an accelerated approval for QualSADI for SOD1 ALS as a reasonably likely surrogate. That really was based on that totality of evidence, all of those different pieces coming together. And I think one of the key challenges in the qualification paradigm is that regulatory confidence isn't really allowed to build over time in a predictable and transparent way. There is really that limited transparency in how confidence can scale. And limited clarity on how you can expand, as I mentioned earlier, on the context of use without really effectively starting over. And that's really where I see the opportunity to modernize qualification, to kind of allow that confidence to build and expand on and advance the biomarker science so we can translate more reliably the meaningful benefits to patients at scale. So moving and probably, as I mentioned before, connecting the product-specific pathway with this qualification that really does kind of advance the science in a more public and transparent way.
I wanted to add in on that, Lauren, just because FNIH has been involved keenly with a lot of the neurofilament light work, and we were doing there. So not only to augment and second exactly what you were talking about, but in that scenario where that goes through an IND process to get that level of evidence to move that drug forward. Now, is that body of data and evidence, along with other more exploratory or previous data, why can that not be brought together to accelerate that through the qualification program and process? Do I have to go reinvent a project to try and do that and pull that data set? Where we have been successful, not necessarily through full qualification, is where actually FDA has been the originator of things. We've done things in hospital and community-acquired bacterial pneumonia where we brought in symptomatic endpoints. And actually, FDA was the one doing the analysis with us and in partnership on our team. And they brought that forward and really had approved multiple drugs, expanded labels of different antibiotic drugs because of the different evidence that they brought through their IND program into what we were doing and some other PRO work and qualification work. Didn't go to qualifications, but really advanced drug development, drug approvals because of that coordinated effort. That type of partnership is really some of the next steps with FDA. We got to get back to that. Use those case study models where we can be successful together and take parts and pieces of data from different efforts. Bring that together. We might be able to accelerate some things through the qualification program and really advance quickly.
Yeah, I couldn't agree with you more, Steve and Lauren, because each one of us reinventing the wheel was not the intention of this program. And it's not, at the end of the day, serving any of us or serving patients. We should not be wasting resources on recollecting data or reestablishing a level of evidence that we all intuitively know has been established. And those challenges only become greater as biomarkers and drug development tools become more complex and they start using sophisticated technology and methods like AI and machine learning. And we know tech cycles inherently are faster than drug development cycles. So if we were able to use technology to detect disease earlier or get a finer gauge of the symptoms of disease or start measuring what matters to patients, this is what we're all collectively aiming to do. So this excitement, Mike, that you mentioned around AI and biomarkers is exactly that. We want to build on what we know, both from a biology perspective and from the tool perspective, because now we can use these sophisticated algorithms and get to treat patients faster and better, right? So, and to your point, while the tools might change, the biology remains the same. So we shouldn't have to reestablish what's already known just because you're using a different tool. So, Katie, you're best positioned to speak to this. PathAI has done some truly pioneering work in this space. So, what would a more fit for purpose approach look like as we start bravely going into the digital world and embracing it in medicine?
Yeah, I'd love to answer that. And so, to your point, a lot of the ways we measure biomarkers these days are becoming more and more inseparable from the methods that we use to measure them. We're finding that we can get more reliable, precise, and accurate data, which can allow for example, higher signal to noise ratio in detecting these endpoints. As we're going through the program, though, these technologies are evolving. And so we do need to separate out the tools themselves and the standards that are required for validation of those tools analytically, and for a particular context of use. I can use, our AIM-NASH AI Assist tool as an example of that. We're not creating a new reasonably likely surrogate endpoint. We're creating a better way to measure, a more reliable way to measure one that is an imperfect gold standard. And so I think in the beginning, we need to, during the qualification plan phase, we need to start incorporating in something like a predetermined change control plan that is used in the diagnostic space that talks about with anticipated changes to necessary inputs like scanners, how we will handle those updates, how we will detect major changes as we use this in a trial, and then how we will revalidate accordingly if there are changes detected, both in the instrumentation used, but for example, the clinical trial population itself. And that allows you to align with FDA up front on what those changes are anticipated to be, how you will validate them, and so that you don't have to go through the requalification process all over again, but move efficiently with the product life cycle, and already have those validation plans laid out in the beginning. And then I think the second thing that I would say is as we are providing clinical validation evidence of these tools, within a particular context of use, say a reasonably likely surrogate endpoint, we would love to mechanism to evolve that context of use. For example, we have tools that go beyond ordinal scoring, that can now provide with more sensitivity and granularity response data that we could now validate against endpoint data and get so much more out of these images with quantitative technology than we are right now with patients, where we're seeing lots of false response rates due to reader noise. And so I think there's improving our measurements for the current biomarkers and then defining within a program what the evolution would look like for anticipated changes and sort of the next step to basically perhaps evolve the biomarker itself to come up with better imaging-based endpoints. And I think a pathway to evolve is really inherent, needs to be built into the qualification plan process up front.
Thank you. What you're saying is we need to think through both the technology changes that enables us to change the methodology we're using to capture the measure, and a way in which we have a new measure itself being validated. Both are important and both are needed.
Yes.
And in reality, the evidence that we develop often comes in stages. We don't have an aha moment where we have the soup to nuts of it laid out. We build, as they say, we stand on the shoulders of giants. We do. And we build by learning iteratively, because that's science. But as I understand it, and as we all understand it, the pathway is almost disconnected from that reality of science and expects all of us to present the FDA with a bolus of data that answers all of these questions. So, one of the things that we have all proposed in our white paper is this concept of a tiered mechanism that would allow you these degrees of freedom. So if you are just changing the technology that is being used in the tool to capture a measure, there is one level of evidence that would be needed. But if you are proposing a wholesale brand new biomarker that has never been seen before, but is based on new biology, new evidence, that would require a different level of evidence generation. So, Lauren and, well, anyone, speak to me a little bit more about the details of what we've proposed and why that would actually be a better incentive for people using this pathway.
I'm happy to get started and then hand it off to anyone who'd like to chime in. But yeah, I mean, I think as you described it, Rasika, really what we have now in the qualification model is this single end stage decision, right? And it's like all of your eggs are in that basket. And I mean, Steve spoke to the amount of time and resources it takes, but really from a regulatory endorsement perspective, it's an all or nothing, right? So what we're proposing in this kind of tiered or more life cycle based approach is really, as I alluded to earlier, about reflecting more how biomarker evidence does get generated. You know, we've talked about the technology element, the biomarker science, the mechanistic data, but really what we were hoping to be able to modernize is, are there different stages of recognition and regulatory endorsement as that confidence builds over time, as evidence matures, that we can build into the existing qualification system. And, you know, we certainly understand that concepts like context of use need to be specific, and we need to kind of work within that framework and sort of how the statute is. But can we sort of create these milestones or tiers of evidence, you know, 1, 2, 3, 4, about how that totality of evidence can evolve? So, you know, the technology validation, the mechanistic data, et cetera, and so forth. And so, you know, certainly what I'd really love to see is BQP to become less of that single determination and more durable and predictable and reflecting how those biomarkers are actually developed in practice. But I'd love to hear from some of our other panelists.
Just a quick jump. I'm sorry, Mike. I just was going to say,
go ahead, Steve. Yeah.
What we were just talking about too is on the NASH front, on the MASH front, and where, you know, we have started programs there where we have quickly accelerated to come up with novel, non-invasive diagnostics. But that body of evidence on a longitudinal assessment quickly changes the context of use or for what's needed in the trial, going after a reasonably likely endpoint and that project. And when you start talking about where I want to bring in partners that are willing to support shared risk, shared resources, they want that flexibility and they want that ability to flex as needed as the program matures, as the technology matures, and as the use of that biomarker. For me to go and restart a brand new program and a project and buy someone in to go after a different context of use and duplication, really not a good model for the consortium building and for partnership. So I wanted to throw that in there, and Mike, that may tee up.
Yeah. No, I was just saying, I mean, I think that we see this concept of tiered kind of regulatory approvals in other spaces, right? We have conditional approvals for drugs and accelerated approval. And, you know, I think something along those lines would make a lot of sense here. One can imagine having, you know, a conditional approval or a different level of evidence for secondary endpoint, where the agency is kind of providing incentives for us as sponsors to use these more often. Because we have some regulatory, assurance that it will be valued within the context of the submission, and then that would generate more data over time. And the agency, it doesn't have to be the only endpoint. The surrogate endpoints of course, is the higher bar, but one can imagine having kind of lower bars along the way, such that we have now we're incenting our sponsors to kind of use these things more often. Same thing with PD biomarkers. Lots of things can start as PD biomarkers, pharmacodynamic biomarkers. We haven't necessarily linked them to outcome, but we understand the mechanism, and analytical validations can be perfectly in line with something like that. Because it's not going to be the only thing that the agency is going to be making their determinations on. So when we talk about things like confirmatory evidence, for example, there could be other thresholds for that for particular biomarkers. So it's really not hard to imagine having different thresholds of evidence depending on how we want to use it. And I do think that the qualification program had a one size fits all, not to their fault. Again, I think just the qualification program coming to existence was a huge win, and we all have to kind of acknowledge that. But it certainly, I think at this point is a good time to sort of for us to reflect and see how can we make it better. Yeah.
I agree. One other point I'll throw out is, when the qualification program was conceived, the concept really hinged on a single entity seeing this process through from, in submitting the letter of intent to getting it fully qualified, whether that is a sponsor or a consortium or, whatever it might be. And the reality of drug development today is very little of this work is carried out by a single entity. And just in our world, you could see one sector, which is the vendors who are developing these tools or diagnostics, which is very different from the folks who are working in the front lines of the clinic who have access to the clinical outcome data, who are able to show you clinically why there is value in measuring differently or developing a new tool. And the incentives are not quite aligned. And when I say incentives, I'm not talking about dollars and cents. I'm just talking about being able to see this process through until the very end, which is what the pathway currently conceives. So one possible advantage of having this tiered expectation is also that each entity is able to come in, contribute what they can, and then leave it for the next entity to pick up and start building on it. So that's another, I think, important factor, which is a design concept we're introducing possibly into this remodeled qualification pathway we might have. I want to pivot really quickly and start asking you, do you think any of what we've proposed is anything that we've proposed is something that FDA can pick up and start implementing today, or do we all have to wait for congressional action? Maybe I'll start with you, Katie, since you didn't get a win in the last one.
I do think so. I do think there need to be some changes so that there's more predictable timelines and resources. But I think there's a lot of opportunity within the current framework. For example, to build out the process during the full qualification package review near the end of it, like we do in the diagnostic and drug space, to collaborate on that published guidance at the end so that we can really make sure it's clear to sponsors then who are going to use the biomarker, the qualified biomarker, exactly how they should be using it in their clinical trial workflow. And I think at this point that it's a little bit of an unknown near the end of that program. Things are published, and then you get a lot of questions coming in, both to me and my company, as well as FDA, from various sponsors asking those same questions. And it's really just made me realize that we can provide more predictable, we can craft that language collaboratively so that it's more usable. Those guidances are at the end are more usable by sponsors who are about to incorporate these into their trial design. So I think that's one thing that we could improve upon right now without changing any statutes.
Yeah. So then you brought up statutes. So let me jump into, do you think Congress needs to take action? And if they do, what would maybe be the single most important thing they have to do? And I'll throw it open to anyone who wants to speak up.
Yeah, I don't think, I mean, it's always, in many ways, I think congressional action would be welcome to the extent that that can provide, additional resources or to codify, I think, in a way that we have in sort of drug development, things like user fees, for example, which are more codified, as also the timelines for review and everything is a little bit more codified, I think, than we see right now in the program. So I think that we're talking a lot about now, do we need to do that or not? That's I think a question in discussions with the agency, but I do think that there are opportunities for congressional action to sort of help sort of facilitate some of the changes that we proposed in the white paper. Yeah.
Maybe just to add to Mike's comments and to Katie's, I do think a lot of what we've discussed, the context of use expansion, et cetera, are very much possible. And even the concept of this tiered framework, there might be nuances of that are helpful to revisit the statute. For those of you, I spend my free time reading statute as a regulatory policy person. I know not everyone's as close to it as some of us. But honestly, just revisiting it before this conversation, I think there actually is a lot of opening in terms of the interpretation in the statute, and a lot of what we're discussing, even in this concept of breaking this down into a slightly more nuanced and tiered approach, is possible within FDA's current jurisdiction.
I may add that there is also existing mechanisms that's already funded through FDA, whether that needs greater congressional dollars and adding money. I don't want to always say that you need more money, but even I think about the FDA support of the BMDR, the biomarker data repository through the Critical Path Institute, where they're looking at safety markers and accumulating that data. When we talk about all these different data sets and data resources, there are existing repositories and data collaboratives that are there, already funded, already established, that may be able to accelerate some of these biomarkers. So maybe it's also kind of a look internal to what's already existing, and are we pulling those levers? Are we using what already exists in the maximized way? And if there is some additional support and funding and effort through congressional action to further support that or better support that in a defined way. We're all biomarker folks, right? We realize the importance of that in accelerating drug development, accelerating treatments, and understanding a disease. So if we're going to really invest in taxpayer dollars, NIH, FDA, all the other efforts that we do that, then this has to be part and parcel with it. You have to work on this validation, adoption, qualification type of efforts where appropriate and needed in the space. And you got to support that. That has to be a priority. It's might not be bullet one, but it got to be in that top set of bullets of where we need to direct our efforts.
Thank you, Steve. And thank you, everybody. This has been a really, really great discussion. And I'm not just saying that because I'm a policy geek. I want to also thank friends, Jeff, Ryan, Bernard, who did a lot of the work, Ellen always, for convening us. And I kind of want to take moderator's prerogative and stand on a soapbox and say a few things now. We've been talking for about an hour, discussing a very esoteric issue: how to improve qualification of novel biomarkers and drug development tools. And that's a mouthful. And most people outside of our world are completely unaware of what we're talking about or what these things are. It's a really niche topic, but it is important that we address it, because what we're actually talking about is how can we improve drug development? How can we better serve our patients, some of whom have rare diseases that do not have established biomarkers or endpoints, or others where we have found a better way to measure their clinical response or outcomes. As much as this seems like a theoretical problem or an operational problem or an administrative problem, it very much is not. Unless we resolve this issue, we will not be able to realize the potential of our investments in basic research and translate them into cures for our patients. So I want to thank you all, thank the audience for listening, and now I'm going to turn it back to Jeff.
Thank you, Rasika. And thank you to all of the panelists for today's discussion. I also want to thank Senator Kaine for his opening remarks to kick off today's meeting. I'd like to thank all of you for joining today's briefing. And as a reminder, the full report describing opportunities for enhancing the FDA Biomarker Qualification Program is available through the link in the meeting chat and on our website. If you have further questions, ideas, or interest in moving the concepts discussed today forward, please don't hesitate to reach out. Thank you, and have a great day.